I'm going to introduce Dr. Patterson-Kane. You remember Janet was here a few months ago and chatted with us about laminitis, and she did such a great job. Liz and I asked her if she had any other talks she could give, and she said, how about canine mast cell tumors? We said, yay, because that's, as you guys know, one of the girls' 4 main cancer endpoints. So it's a timely discussion. So I will let Janet take it away. Thanks you so much again for coming.
Okay, so canine mast cell tumors. So I'm just going to go through why they're so important, kind of what happens when a dog goes to a vet and has a mast cell tumor diagnosed. And then I'm just going to talk about a bit of research that I personally have been involved in. I thought I'd start really basic because I think there's a lot of language around cancer that actually even I see veterinarians being confused by. So I'm just going to start just with some really simple things because we throw these words around without really perhaps understanding. So I've put some chihuahuas up here to keep your attention on those definitions. So a neoplasm is a mass formed by cells that either grow abnormally and or they're not dying when they should, often both things. So that's what we call a neoplasm. When we use the word tumour, that means the same thing. So neoplasia, neoplasm, tumour, all the same thing. So if we look at neoplasia, neoplasia can be benign, neoplasia can be malignant. So a tumour can be benign, a tumour can be malignant. Things that are benign tumours, they often grow slowly, but they can still cause a problem. So if you have a benign tumour inside your brain, that is a problem because it's occupying space that you can't afford to lose. If you have, for example, you see sometimes dogs with huge lipomas walking around, that's a problem because the skin overlying it can start to ulcerate. So it doesn't mean it's not a problem, but there's a difference between that and having cancer. So cancer is a neoplasm, but it's a malignant one, okay? So just going back, a benign tumour, a benign neoplasm is not cancer. So cancer is the focus of oncology. So we see all sorts of things in animals that are benign tumours, they're not cancer, and you'll find members of the public get very confused about this. So you'll start talking to them about a benign tumour in their dog and they'll be panicking because everyone thinks it means cancer, but cancer is something very specific. Okay, so if we look at a benign tumour, there's a diagram here, they tend to just be nice, well circumscribed, and they just gradually expand and compress the tissue around them. Or they might sit there and do nothing at all that's very noticeable. Cancer is different. So cancers are often very irregular, they invade the tissue around it, and worst case scenario, they develop the ability to go to another place in the body. So that's what we mean when we say metastasis. Okay, so we've got neoplasia. Neoplasia can be benign, can be malignant. If it's malignant, it's cancer. Cancer invades tissues around it. Cancer can spread to other parts of the body. So that's what we're dealing with our cancer awareness. Okay, so going even more basic now. So what is neoplasia, either benign or malignant? It's basically a genetic disease. So we've got changes in the genes that are controlling how the cells work, usually particularly how they grow or how they die. These changes might be inherited. So we see lots of cancers that are linked to particular breeds of dog, for example, or breeds of cat or certain families. Or they might be acquired, so tobacco smoke. Pets can get cancer from tobacco smoke, just like we can. Ultraviolet radiation, so squamous cell carcinoma in animals is a very well-known example. Now, it's usually not just one mutation, it's kind of an accumulation. So you don't just suddenly have cancer. This is developing over a period of time, and you usually need more than one mutation. So it's happening over a period of time, and actually as it gets worse, you're more and more prone to getting more mutations in those cells. Right, so we start off with something quite innocuous. It starts to look a little bit not like it should be, and we'll see this as pathologists, things that are not quite cancerous yet but are getting that way. Really starts to look a lot worse, and finally full-blown cancer. And so, I mean, I've given you gremlins as an example, but actually when I look at slides I can see this. So you'll see normal cells, cells not quite so normal around the edge, and then you'll see the really nasty cancer cells in a malignant tumour. Okay, so what are mast cells? So they're a type of white blood cell. You know, we have red blood cells, we have white blood cells. White blood cells are involved in immunity and inflammation. Mast cells have granules, so you can see up here, you'll see all these purple granules. Those are the nuclei in the middle there. And in those granules, they're very rich in histamine. You'll have heard of that. So we take antihistamines for allergic reactions. They contain heparin. You might have heard of people taking heparin to stop blood clotting. Okay, so that's what's inside those. And we best know them for their role in allergy. So this is a classic dog with a bee sting before and after, tragic swelling here of the face and ears. But they're actually involved in all sorts of things, and this has been recognised more and more. They're involved in wound healing, they're involved in defence against infection, so they're very important cells. In the skin, and mast cells are you know, in various tissues, but in the skin they hang out around blood vessels so that they can monitor what's going on there, take up some molecules that they need if they're functioning in an allergic response or something else. So if we look here, on the side here, okay, so this is a blood vessel, and those are the mast cells. And so this is quite normal. You look at normal skin, you just see a certain number of mast cells just sitting there. It doesn't mean that it's a mast cell tumour. Okay, so how common are they? It's really hard, as you probably know, with animal populations to try and work out how common something is. It's really hard to get that information. In one UK study, they thought about 129 out of 100,000 dogs, but this was only insured animals. It doesn't sound like much, but it's really hard to get a handle on what that actually means. There was another study where they looked at dogs just going to veterinary practices in England, and they found that 0.27% had a mast cell tumour. Now when you think of all the different things that dogs go to the vet for, that seems like actually quite a lot just for one tumour. And then there's all the people that don't take their dogs to the vet, and we never really know how common it is. However, by the time we get to the point of we have a dog with a tumour, what is the diagnosis? We're looking at, in various studies, between 7 and 21%. So if you think of all the skin tumours we must see on dogs, that's an extremely large number. And there are breed differences. So we know golden retrievers are prone, and we know boxers and pugs and all sorts of breeds are prone. Just to give you an idea, when I worked for IDEX in a diagnostic laboratory, I had to do about 60 biopsies a day, and on average about 15 of them would be mast cell tumours. So if you kind of multiply that by all of the pathologists sitting around looking at dog tumours, that's a huge number. And there's probably another 10 or 15 that was soft tissue sarcoma, which is another very common one. So of all the cancers we're dealing with in dogs, mast cell tumour is the big one. Mast cell tumour is also more likely as dogs get older, and of course cancer in general is, and it's more likely in a dog weighing 20 to 30 kilos, I'm sorry, I'm in the metric system, versus a little dog like this one, so the risk of mast cell tumour would be less based on size. So there's all sorts of these different factors coming into it as to whether a dog is prone or not. So what do they look like? A lot of people seem to think they can diagnose things just by looking at them, and I'm actually very reluctant. There are very few things I would look at as a veterinarian and say, oh, I know what this is. So they can look like anything. They can be tiny, they can be enormous. A lot of dogs have multiple ones, so 10 to 15%. You're sitting there getting the biopsy and they've taken multiple masses off. They can be anywhere on the body. They can be in the dermis, which is just under the surface, or they can be deeper in the fatty tissue. And they do behave a little bit differently there. So they can be anywhere. If your dog gets a lump, please get that looked at. You can't assume, even if it looks like a lipoma, right? My dog had a lipoma. I had that aspirated because a mast cell tumour could still be in there. So it's important to tell people that they actually need to have these things diagnosed. You can't just assume that something is benign or something is a lipoma. Okay, so in up to 96%, so pretty much most cases, the diagnosis can be made at the hospital on the table by sticking a needle into it, pulling out some cells. Cells get put on a glass slide. We put a stain on. This is one of those things you don't need to be a specialist pathologist generally to make that diagnosis. So we can see all these round cells here with granules in them. So when I'm getting cases sent in, most of the time they've already made the diagnosis before they take the tumour off. So it's not really a shock diagnosis. And it's important that they know that before they do the surgery, as I'm going to show you. Okay, so the problem is that all mast cell tumours are potentially malignant. So they need to get the mass off completely by surgery because it will be invading the surrounding tissue. It is a cancer. But we're also concerned about whether it's spread to other tissues because they can do that too. So it is a cancer, it can invade locally, it can spread to other places. Where does it go first? Well, they first go to lymph nodes, which some people call glands. So you know when you get a cold, your glands will be up here. Those are your lymph nodes. And different lymph nodes drain fluid from different areas of the body via lymphatic channels. And tumour cells use those channels to spread to the lymph node. So that's the first place we'd be worried about. And that determines which ones they should look at. It's quite surprising how recent this piece of work is, 2014. Because veterinarians will often say, what lymph node should I check? It was on the right front toe. And as you can see, it's all rather complicated, but you can see here on the hind limb, see the front of the leg here, but then that goes up here, and then there's a different region here. So people need to think about where that could possibly go. And there's a bit of individual variation. A lot of veterinarians don't bother to assess the regional lymph node, which kind of annoys me as a pathologist. Certainly in human medicine, they would be doing that. So it's quite important to know if that has happened. And sometimes they might do something like an abdominal ultrasound if they think that it might have gone and spread further. Okay, so if at all possible, they're going to try and take this tumour off surgically. It's not going to be left there. There's not really a therapy you're going to do without taking that off. They generally aim to get 2 or 3 centimetres around it. So here's our mass here. You can see they've prepped for surgery. They want 2 to 3 centimetres around here, here and here, and at least 4 millimetres deep to it. So we'll get a piece of tissue like this. That's sent in formal and fixative, that's sent to the lab for the pathologist to look at. It doesn't matter they've already made the diagnosis, there's a lot of things that they can't do just from those cells. Sometimes they might dip it in ink, which is really useful, because when we finally cut that onto the slide, we can see the ink around the edge of it, and then we can talk to them about whether those margins are clear of tumour or not. So what do they want to know? Well, they want to know, have they got it off? Because if they haven't completely removed this tumour, it could come back, and it probably will. So this is the big concern with them sending it in. So this is what we would do. So we get this piece of tissue, and we're making a cut across here. So we can look at this margin, and that margin, and this deep one here. Then we come out this way to get this other margin here, and this one here, and then this margin. So we're not looking at the entire tumour, it's not possible. Each section is about 4 to 5 micrometers thick, and a micrometer is 1,000th of a millimetre. So you're just doing the best you can because you can't cut that entire thing in. You would have thousands of slides. So we're just trying to hit those margins so we can give them at least some idea of whether they've got that out. And now, I mean, I've been working digitally for some years now, so we have it on the screen and we can actually measure how far that is to the margin from the edge of the tumour. One of the problems, though, is that malignant tumours don't really look like this, so you'll have all these kind of little fingers or tendrils of cells potentially going out into the surrounding tissue, and you might not necessarily get those in the sections that you've looked at. So there's never this 100% guarantee. But if you've got margins of several centimetres, that's generally good, and you've probably got the tumour out. So what do we need to know? Well, you would like to know, how long is my dog going to live? Now, my dog has had this mast cell tumour. How likely is it actually did metastasise to a lymph node or further? You'd want to know that for your dog. How should the dog be treated? And we have this huge problem with mast cell tumours, and that is they vary enormously in how they behave. Some of them look really very benign. We take them off, nothing else ever happens. Others of them are incredibly malignant and they spread all over the body very quickly. And that's the problem with mast cell tumours. There's just this range of behaviour. And we need to know, if that's your dog, whereabouts in this range is it, and what do I need to do? Okay, this is a very complicated diagram, but it's just to show you the very confusing place we are in right now with these mast cell tumours. So the first grading system that was developed was in 1984, and it's called the Patterson-Kane. I really don't like this grading system, but a lot of practitioners are still hanging on to it, and so you hear it talked about all the time. So we have grades 1, 2 and 3. You don't need to know all of these words, but basically a grade 1, it's very superficial in the skin, and the cells kind of look more like normal mast cells, so that's what we mean by well differentiated. Down to grade 3 that tend to infiltrate very deeply. A lot of them don't look like mast cells at all. Sometimes we have to do special stains, and they grow very rapidly. So this was the system that we had. I'll go into the next slide. There was a lot of problems with agreement between pathologists and what this actually did mean. And so in the last few years, another paper was published. This is the Coop grade. We've just got low and high. It makes it easy because we've just got low and high. And it's based on a number of things, including mitoses per 10 high power fields. And mitoses are things that we see when nuclei are dividing. So we're counting them, and it gives us some idea of how rapidly these cells are proliferating. So as a pathologist, it's easier because it's more clearly defined. So we're counting these fields under the microscope, and we're counting different things, and we're classifying them as low or high grade. So what does this mean? Obviously, if you're up here, low grade cupule, grade 1 Patterson-Kane, it's kind of a low potential to spread. You can probably just surgically remove that. The prognosis, which means what is likely to happen, is probably good. If you're right down this end, your high grade cupule, your grade 3 Patterson-Kane, your prognosis is probably poor. It's probably quite likely to spread or have spread. We'd need to consider surgery, perhaps radiotherapy, perhaps chemotherapy, perhaps other sorts of treatments. The problem comes in here, in the middle, which is where most of them are. Very few of them are actually low grade or really, really high grade. And so there are all sorts of options. Prognosis could be fair to guarded, you don't really know, and that's where we are. So we can't make these grades off the cells that we just pulled out of the tumour and put on a slide. We need that tissue to be sent in. We're looking at whether it all got excised, but we're also looking at the grade. How is this likely to behave? So, it's very important that a grade be linked to what would happen clinically. But remember, we're dealing with population statistics, so when you're looking at your individual dog, this may or may not actually apply. So with the Patnaic, you can see survival longer than 1,500 days, and you can see it going down very rapidly when we get to grade 3. If we look at the grade 2 specifically, you may as well flip a coin as to what's going to happen, which is not where you want to be with the grading system, or when you're deciding what to do with your dog. If it's 50-50, which is, and most of them are grade 2, that is not a useful grading system. And look at the agreement between pathologists for those grades. This is not good enough. You know, we cannot have pathologists disagreeing 30-40% of the time. And part of that is because the system wasn't very clearly defined, so we all came to our own opinions about what we think are in these different grades. Coop makes it easier, but partly because there's only 2 things, so it's easier to agree if you've only got 2 options. For the low grade, we've got quite a long survival time. For the high, not so much. Very high pathologist concordance, which is absolutely fine, but it doesn't remove many problems with grading, because there are dogs graded as low that turn out to not be, and there are dogs graded as high that turn out to not be. So while it's useful, it's not solving the problem. Okay. So I'm just going to go into a piece of research that I've been involved with, and I'm going to talk about biomarkers. I don't know if you've noticed this in the literature or the news or things that people write about cancer, but this is a big thing in medical research now. Everyone's trying to find biomarkers that tell you what cancer drugs you should be using. And biomarkers are going to be just as important, and already are, in veterinary medicine. So we have some biomarkers for mast cell tumour, but they're very difficult to interpret, and they kind of conflict with each other a lot. So you can have all these things done and still, they're not great biomarkers, and they need more work done on them. So biomarker could be telling you what the risk of a disease might be. So a preventative thing like the BRCA gene for women with breast cancer. We now know, some women have their breasts removed when they find out they've got that gene because the association is so high. I think we'll get to that point with both veterinary and human medicine. So they're now talking about tests that will screen the blood for molecular markers of cancers that haven't yet spread. That's predicted to come in the next few years. So this is the sort of thing that veterinary research should also be looking at. There are other biomarkers that are diagnostic. We use them all the time. So these are very common things that we use. Tell us, is this a mast cell tumour or isn't it? What is this thing? And then there's prognostic, which is what we're looking at here. So your dog has mast cell tumour. Is there something we could use? The dog's already got the disease to tell us more clearly what's going to happen. So some biomarkers will tell you what will happen if no therapy is given. So that was kind of what we were looking at just then with the grading. That's kind of saying, if I didn't treat my dog and it was low grade, how likely is it my dog is going to live longer than 2 years? That's the sort of thing we're looking at. What we really want is which therapy is the best one to use. And we're not really there with mast cell tumours with that. And what we'd also like to know is if I gave that therapy, how likely is it that this would respond? So this is all very ideal. That's what we want biomarkers to do. The reality is, even in human medicine, there aren't that many very useful biomarkers clinically. So the biomarker I'm going to show you, we're probably at this stage at the moment, what will happen if I didn't give any therapy. Okay. So the biomarker that I've been working on is called P62. It's a very boring name. It's not an exciting name, but it's a very exciting protein. So if we think back to the mast cell, so it's just a diagram. So this is our mast cell, this is our nucleus, this is our cytoplasm. For those of you who don't know cell biology, the nucleus is where the DNA is, so it's kind of like the brain of the cell, and the cytoplasm is out here. So most things are kind of, so the DNA is transcribed, and then proteins and things are made out here in the cytoplasm. So P62 is a protein, and it moves very quickly backwards and forwards between the cytoplasm and the nucleus, which is a very complicated process, and it's monitoring both compartments. It's monitoring those compartments to find damaged protein. So we have damaged protein in our cells all the time, and it identifies that protein and it takes it to either be broken down completely or recycled and used again. Now, in cancer cells, we have a lot of damaged proteins. Cancer cells are out of control, particularly the more malignant ones. So how P62 moves around finding damaged proteins and helping to deal with them tells us where the cell is stressed. Now, this is very useful. It tells us what the cell's up to, but ultimately it can tell us where we should target the therapy. Because, for example, chemotherapy is targeted at certain processes in the cell. So we got 66 cases of mast cell tumour from IDEX. This was a few years ago now. And we stained them for P62. The co-investigator in my lab group had been looking at this protein in Hodgkin's lymphoma. And it was very complicated. And I kept saying, you should look at this in mast cell tumour because, you know, it has a lot of granules and protein. I thought it would be useful. And finally she got sick of looking at it in lymphoma and said, okay, get some mast cell tumours, let's have a look. And we were amazed at what a good marker this was. I mean, it was better than I had actually ever expected. So in 14%, a small percent, we just saw P62 in the nucleus. Puncta means just little clusters. And there was very little in the cytoplasm at all. You can see the nuclei here. Now, if you look at a normal mast cell, it's really hard to see any P62 at all. So this is abnormal. Let's have a closer look here. Here's our nucleus, and you can see those puncta of P62, but you can't see it around the nucleus at all. So that was our first pattern. In 44%, so quite a large percent, P62 was in the nucleus, but it was basically completely filling it. And then it was also spilling out into the cytoplasm. You can kind of see the puncta in the background there. So a lot more P62. It's in both compartments now. And in 42%, again, quite a lot of them, it had basically left the nucleus altogether and was just in the cytoplasm. You see how dark that brown staining is there? So we've got three patterns. Nucleus only, filling the nucleus and spilling out into the cytoplasm, and in the cytoplasm only. And there are various processes occurring in the nucleus and the cytoplasm. This can mean, when it's in the cytoplasm like this, that it's been recycled and it's been used by the tumour cells to generate amino acids and kind of fuel itself. But we're not certain. So we proposed it was recycling there, but we don't know. So then we looked at the Coop grade. Now that pattern A grade that I don't like, there was no correlation with that was meaningful. But the Coop grade where we have the low versus the high. So if we look at our nuclear pattern, they were all low grade. If we look at our mixed pattern, most of them were low grade. If we look at our cytoplasmic, most of them were high. But look at these ones here, okay, with these mistakes, because we have mistakes with these grading systems where we think something is going to be low and then we get oh no, it actually metastasized to the lymph nodes even though you said it was low grade. So would this biomarker help us find those mistakes? Similar things have been seen with human cancers. So since we actually did that work, a lot more came out on all sorts of different human cancers and how P62 in the cytoplasm seemed to associate with more malignant behaviour. But nothing was as clear-cut as these dog cases. So we looked actually at other canine cancers, we looked through the literature, but nothing was really as clear as this. So there, and I'm not going to tell you what it is, but there's something quite specific about dogs that makes this a really great marker for this cancer in dogs. And at the time, we decided to culture some cells, and everyone said, you can't culture mast cell tumour cells, it's not possible. And we got some clients to agree that if we would look at the biopsy for them and do all the grading for free, they would let us have the tumour. So we would rush up in a taxi, this is in Glasgow, grab the tumour from the operating theatre on ice down to the lab, take some fresh tissue to culture the cells, and the rest would go through the same procedure it usually did. And we named the cultures after the dogs themselves. So this is Bailey. Bailey had a low-grade Coop. Bailey had a nuclear P62 pattern when we stained for that. So this is the tissue. So these cells look fairly similar to normal mast cells. They're quite small. Their nuclei are roughly the same size. When we cultured them, these cells in the background here are actually fibroblasts, and they lined up on top of them and actually fed off those fibroblasts. And then you can see, it's a bit light in here, but those red dots are the nuclear P62. So we had really great agreement between the cell culture and the dog tissue. And this is Jules. Jules was a Weimaraner with a high-grade tumour. So you can hear the mast cells. See that big nucleus just there? Huge. So they kind of vary a lot in size. They don't look as nice as these ones at all. In the culture, they're all kind of clumping together. And you can see all this red here, which was in the cytoplasm. And so we did actually study some drug effects with those cultures. And, you know, every culture was its own thing. Every mast cell tumour is its own thing. So if you're applying population statistics like we saw with those grades, you're not really providing what we would call personalised medicine to that animal, or what they're now calling precision medicine actually in the human field. And so people are starting to get these sort of precision medicine treatments, and they're going to be wanting them for their dogs, because dogs are seen as family members. So we need a better biomarkers linked to treatments. There's just so much work to be done with something that we see every day as one of the most common things that we deal with. Okay, so take-home messages. Mast cell tumours are common, skin cancers in dogs, probably the most common one. Pathologists see a lot of them. So we look at these every day, apply these grading systems. Personally, I don't feel like I'm being very useful because we're not able to direct therapy or really absolutely tell people what's going to happen. And that's because they're so variable, right? So if you have a lymphoma, you pretty much know that is not great. If you have a mast cell tumour in a dog, it can be anywhere from fine, just excise it, to terrible, metastasising all over the body. The grading systems are, in my opinion, very crude, and I think everyone recognises that, who's involved with it. It's really just giving you a fairly vague idea, unless you're falling right at the very end of high or low grade. There's a lot of uncertainty. So we need better biomarkers, and P62 is a very promising one. And this is very typical. So then that covers all the points, and we're up to questions. Thank you. Hand up the back there.
How long can a tumor sit in formalin and still look at P62?
Yeah, I mean, that comes down to, so what we're looking at there when we're staining P62 is we're actually using an antibody against P62, and that antibody is via another antibody linked to our brown marker. So if you leave tissue in formalin for too long, usually or often what happens is it changes because of the fixation and the antibody doesn't recognise it anymore. So you generally don't want to go above about 48 hours. That's not to say it still wouldn't work, and you'd have to do a study to actually work out what that was. But in general, you wouldn't want to take a tumour, been sitting somewhere for a week and decide that you were going to use that. But that's pretty standard. And with diagnostic labs, veterinary ones seem to work much faster than human medical ones. So, you know, the turnaround time once you've got the tissue is usually 1 to 2 days. Most labs I worked at, you receive the tissue one day, you're looking at it the next, so it's pretty rapid turnaround.
So I have a question for everyone, Janet. Can you talk a little bit, even though we're talking about dogs, about mast cells in cats?
Yeah, sure. So, well, if we look at mast cells tumours in humans, for example, they're very rare. So, you know, if a medical pathologist saw that, they'd be really excited because they just don't see these things. Whereas a veterinary pathologist, that's one of the most common things. Mast cell tumours, and I'm just saying this is just thinking about what I see as a pathologist, it is one of the more common things from cats, but they tend to just be locally invasive. So it's more of a local problem, and we're more concerned with, did they excise the tumour, looking at the margins. They do tend to get them around the head, around the face, and they can be multiple. And in certain breeds, it's a real problem, but it's not the same thing as when we're dealing with dogs and worrying about whether we need chemotherapy or radiotherapy. Horses get mast cell tumours. They're not particularly common. They're usually benign. Again, they're locally invasive, and they can cause problems with trying to get the whole thing out. So yeah, we're seeing them in multiple species, but there's a different thing to think about with each species, and that is actually a reasonably common thing that a veterinarian or a veterinary pathologist has to deal with, that someone in the medical field doesn't have to think about. You know, they don't have to know those different behaviours in different species.
So have you tried P62 in cats?
We did start a study. We haven't actually run it yet, but there is something specific about P62 in dogs that is different from other species, including humans. So it may turn out that it is only usable in dogs. So we can't always take a marker from the human medical field and think, oh, this is going to be very useful for cat, dog, horse, or vice versa. So we have to develop our own biomarkers as well. We can't just assume that if we developed a mast cell tumour that it would automatically be usable in a human cancer, because I haven't seen those same patterns convincingly there. There was one paper on prostate cancer that did seem to show that, but then someone debunked it in a later study. So, you know, there are things that translate, there are things that don't.
Anybody else have any other questions?
Does this occur at all in wildlife?
Mast cell tumours? Yes. Yeah. I mean, I think in various species you'll see various things that we see in domestic animals, but you know, they don't tend to live as long. We don't tend to look at them, but I've certainly seen things in zoo animals as they get older that are very similar to things we see in other species. I'm not aware of anything with that range of behaviour as in the dog.
I think we're done.
Thank
So
you.
much.
Okay.